everyone, my name is Paige Wilson. Today, with Emma and Tilly and Zoe Bart, we will present the evolution of vaccines. Genetic engineering is using biotechnology to modify the genetics of organisms. Since DNA technology was discovered in the 1970s, research in this field has come a long way. Recently, genetic engineering has been the latest topic in food and medical industry. For instance, in food, there are possibilities of genetically modifying plants and animals to produce faster or the ability to use less pesticides in plant growth. In the medical field, some ways genetic engineering is used is to mass produce insulin and treat infertility. Our group decided to read Enhancing Evolution: The Ethical Case for Making Better People by John Harris. He begins by discussing the ethics behind the modification of human enhancement. He then gives arguments on why human enhancement would be good for society. He believes any enhancement, regardless of the harm or possible outcomes from modifying humans, will enhance society. Harris then gives examples of different modifications that could come with experimenting with genetic engineering and human enhancements. For instance, he discusses being able to choose the characteristics that children are born with. He believes this would give children an enhanced chance at becoming successful in life, giving enhanced physical and mental characteristics such as concentration, memory, strength, stamina, and speed. Harris also discusses immunity from diseases such as cancer and HIV and AIDS. With the ability to genetically modify diseases like these, people would have the ability to live longer, healthier lives. Harris believes society has an obligation to use these technological advances provided in the recent years to enhance evolution and make better people.
This is baby Jack. Vaccines help Jack fight infections by introducing a small number of antigens into his body. Antigens are parts of germs that cause Jack's immune system to go to work. After Jack gets a vaccine, his immune system will remember the vaccine antigen and attack that germ if it ever invades his body again. The antigens in vaccines are weakened or dead, so they do not cause illness. Vaccines contain only a tiny fraction of the antigens that Jack encounters in his environment every day. It'll take a few weeks for the vaccines to start working, and Jack may need more doses later to best protect him. That's why Jack's parents are following the recommended immunization schedule, because it is designed to give him the best protection possible from 14 serious diseases.
In order to talk about the history of vaccines, first we need to talk about this guy, Edward Jenner. In 1794, he created the first vaccine to defend people against smallpox. He took fluid from a cowpox blister and scratched it into the skin of an 8-year-old boy. A single blister rose up on the spot, but the boy soon recovered. Again, on July 1st, Jenner inoculated the boy. This time, no smallpox disease developed, proving the vaccine had been successful. In 1884, Pasteur created the first rabies vaccine, the first vaccine to be designed in a lab, and in 1886, the rabies vaccine was used on a human. In the 1960s, vaccinations against measles, mumps, and rubella and the modern tetanus vaccine were created. In 1988, the WHO resolved to eradicate polio. As you can see, throughout the years, the number of cases of polio declined, eventually being eradicated in 1980. 2000s, meningitis C and the human papillomavirus vaccines were created.
Before vaccinations can be introduced to the public, there is an extensive testing process that must be followed with regulations at every step of the way. The licensing process can take years, and after its initial development, there are 3 phases of clinical trials that must occur with human subjects. In Phase 1, the vaccine is administered to a small group of volunteers to determine if there are any adverse reactions to the vaccination. Phase 2 revolves around the effectiveness of the vaccine and includes up to hundreds of people. The purpose of this phase is to gather more data on the vaccine and pinpoint appropriate dosage necessary. The final phase of the trials, Phase 3, expands the number of participants to ensure the overall safety and effectiveness. If all phases are successful, the vaccination can move on to receive a license from the FDA. After this point, the vaccinations can be administered, but the monitoring will not cease. Despite this process, according to the CDC, since not all bodies will react the same way, no vaccine is actually 100% safe. Different viruses present new sets of obstacles that require different approaches when creating vaccines. There is no one-size-fits-all approach that can be taken to create the vaccines that will prevent diseases. Over the years, as technology has improved and advanced, scientists are expanding their methods of vaccine production. Previously, the main source of defense against these viruses had been vaccines that used versions of the diseases that would be of no harm to us as they were weakened in some way. Now there are numerous approaches that don't require using these complete versions of the diseases, although these methods may still be used. An excellent representation of the technological advances in vaccine production today is the ongoing development of a vaccination for the virus that causes COVID-19. According to the National Institutes of Health, Moderna, a biotechnology company based out of Massachusetts, along with other researchers, manufactured a vaccine that uses a virus's RNA to create an immune response in our bodies. As explained by Nature, on the surface of the virus is a spike protein that essentially takes our body cells and latches to them, allowing the virus to enter the cell. But by creating an immunogen that prevents the spike from latching onto the cell, this could prevent the disease. Moderna's vaccination is revolutionary as it uses the messenger RNA from the virus to produce this immunogen in our bodies. The overall goal is that the people's cells who have received this vaccine will continue to produce these proteins to better protect them from COVID-19. Moderna's vaccine has been proven safe in Phase 1 trial, is in a Phase 2 trial, and is soon expected to enter into Phase 3. Because RNA is relatively unstable, there are also vaccinations that use DNA instead. This process requires that the DNA be transcribed into mRNA to make the needed proteins. It subsequently has a much longer storage life, which is important for the production of vaccinations for public use.
Genetic modification would help produce edible vaccinations that could offer an inexpensive and painless alternative of introducing vaccinations into society. This would be especially useful for underdeveloped countries that can't afford vaccinations. Having an alternative, inexpensive way for vaccinations would be life-changing. These edible vaccinations will work by using an antigenic protein produced in an edible plant that is then consumed and absorbed in the bloodstream. Once absorbed, the protein would work the same way as an injected vaccination would work, by stimulating the immune system to produce antibodies against the disease. One of the most important reasons for vaccinations is to obtain herd immunity. Herd immunity occurs when enough people become immune to disease, generally from vaccinations, and make the spread unlikely. As a result, the entire community is protected, even those who are not themselves immune. In regards to the current COVID-19 pandemic, the hope is that the creation of a vaccine would allow the delivery of the vaccination worldwide and create herd immunity against COVID-19 and finally put an end to this pandemic.